Hello everyone, I'm sorry for that problem. So kindly join please. Kindly join so that we can start. Kindly join so that we can start please. Okay, just a minute, that screen is being adjusted. Just a minute, we are starting in 2 minutes. Okay, okay. So we have started discussion of vaccines. So what are vaccines? Vaccines are those substances which acts as a preventive measure. They act as a preventive measure against certain diseases. For example, how do they work? See, similar to diseases, whenever any virus infect our body, as a response to that foreign material which is called as antigen, our body develops certain proteins, and those proteins are in the form of antibodies. Those antibodies will make sure that the action of antigen is restricted and it is removed from our bodies. Fine. So that is the work of our immune system, and that immune system gets triggered when the foreign material or antigens affect our or enter our bodies, right? So that is natural part. But vaccines also use this mechanism. Vaccines also use this mechanism wherein the vaccines try to teach our immune system, if in future if you see this particular antigen, then you will have to develop these kind of antibodies. That is the work of vaccine. It teaches our immune system regarding the future attack which may be possible of that particular antigen of which vaccine we have taken. Fine. So let us discuss this part. A vaccine is a substance that helps protect against certain diseases. They will protect against certain diseases by stimulating. This is very important. How vaccines work? They stimulate a person's immune system to produce immunity to a specific disease, protecting the person from that disease in future. Next, it helps your immune system recognize and destroy the living microbes during a future infection. Fine. So it will help our immune system to detect the microbes which may enter in our body and which may cause us infection in future. Vaccine contain a dead or weakened version of a microbe. Why vaccines are able to stimulate that response by our immune system? Because vaccines generally vaccines are made up of dead or weakened version of microbe. They use that microbe, they use that virus, let's say, to develop vaccines. Fine. There are other major methods of developing vaccines as well, but generally or traditionally, what is used in vaccines use dead or weakened version of a microbe or virus itself to develop or to stimulate our immune system. Next, vaccines are usually administered through needle injections, but can also be administered by mouth or spray into the nose. So generally they are given by needle injections, or there are other vaccines which may be given through oral means as and also by and also through nose. Next, why vaccines are important? Because they are a very important part of preventive healthcare, which should be the focus of any government. Our focus, the government's focus, should be on preventive healthcare, but rather these days most of the governments, they focus on curative healthcare. Fine. That means once person is affected, then treatment is started. But our focus ideally should be on preventive healthcare, because it will help us to reduce the health burden on the person as well as on the state or on the government. Fine. So preventive healthcare is very important, and vaccines are very important part of it. Vaccines are an important part of preventive health care. Vaccination is a way to teach the immune system. What does it do? It teaches our immune system how to recognize and eliminate an organism. Fine. So what is the work of vaccines? They teach our immune system regarding the future attack or regarding the future infection which may be a possibility. Next, the body's immune system helps protect against pathogens that cause infection. Fine. So body's immune system helps protect against pathogens which cause infection, which may cause infection in future. Fine. So most of the time it is an effective system. So this immune system of our body is an effective system, because most of the time if they find any antigen entering in body, they will make sure that that antigen is removed from the body, and body is protected from the attack of that antigen. Most of the time it will work fine, but there are certain viruses which may overpower our immune system, which may lead to strong response by our immune system. Fine. So there are certain viruses which lead to extremely volatile or extremely violent response by our immune system, and that is not the ideal response given by immune system. Why? Because if the response by immune system is extremely violent response, then it will lead to damage to other parts of our bodies as well. And that's why you must have heard about multi-organ failure. Why it happens? Because of the violent response given by our immune system to certain viruses, which is not very good kind of action that immune system takes. Right? So most of the time it is effective. It either keeps microorganisms out or tracks them down and gets rid of them. However, some pathogens can overwhelm the immune system. When this happens, it can cause serious illness. Right? So sometimes what may happen, the pathogens may overpower our immune system, and which may lead to serious infections and hence for such pathogens, what we need vaccines. Fine. So I hope you are getting what vaccines are, why vaccines are important, and what kind of work do they do. How they do that we will be discussing later. But why vaccines are important, I hope you are understanding it. Next, vaccines are important for primary protection. They are a primary protection for any infection. That is, vaccines prevent people getting sick. They prevent people from getting sick, and that's why it is a kind of primary protection for a person. Vaccines are important to control diseases. Why vaccines are important? Because we have already controlled many diseases like polio, whooping cough, tetanus, with the help of vaccines. Fine. So with the help of vaccines we have controlled these diseases: polio, tetanus and whooping cough. Vaccines are also critical to prevention and control of infectious disease outbreaks. So with vaccines, so suppose there are 100 people in a particular city, and if out of those 100 people, if you have at least vaccinated, let's say 60% of people, then you will be able to control the spread of that disease or the outbreak of that disease, and hence your health systems will not be overwhelmed because of such outbreaks. And that is also vaccines are important. They prevent outbreak of certain diseases. They underpin global health security and will be a vital tool in the battle against antimicrobial resistance. Fine. So vaccines, why vaccines are important? This last point is very important. You must have heard about antimicrobial resistance. Antimicrobial resistance is a kind of resistance shown by bacteria, viruses or other pathogens against the drugs that used to cure or that used to act against them earlier. But now they have developed resistance against those drugs. That means those drugs will not be able to address or will not be able to cure that disease caused by those bacteria or viruses. Fine. And that is nothing but AMR. So with vaccines, our dependency on drugs will reduce, and hence it may be the case that the antimicrobial resistance will be controlled with the help of vaccines. Next, how do vaccines work? So I hope you have understood what is the importance of vaccines. Now, how do they work exactly? What is the work of vaccines? Fine. So in order to understand how vaccines work, it is very important to understand how our immune system works. Fine. So पहले हमें immune system कैसे काम करती है वो जानना होगा, and then we'll be able to understand how vaccines work, because we have already discussed that vaccines they try to mimic the response given by our immune system. Functioning of our immune system. So let us discuss how our immune system works. The immune system can be activated by a lot of different things that the body doesn't recognize as its own. So how immune system is getting activated? It is getting activated whenever it come across any material which is not a which is coming from outside and which is not a material of that body itself. Fine. So if it recognizes such kind of material, then it gets activated. These are called antigens. So those materials which our immune system recognizes, they are called as antigens, and because of these antigens it will get activated. Example of antigens include the proteins on the surface of bacteria, fungi and viruses. So antigens क्या हो सकते हैं? Antigens ये proteins हो सकते हैं, वायरस से रिलेटेड प्रोटीन, बैक्टीरियाज या किसी भी पैथोजन से रिलेटेड प्रोटीन हो सकते हैं एंटीजेंस. एंड बेस्ड अपॉन दीज एंटीजेंस immune system gets activated. When these antigens attach to special receptors on the immune cells, a whole series of processes are triggered in the body. Fine. So जब ये immune system antigens को detect करता है, उस टाइम बॉडी बहुत सारे रिस्पॉन्सेस डेवलप करने लगती है. फाइन. As a result, antibodies are produced. So, as a result of those responses, which may be in the form of, whenever infection occurs, we often suffer from coughing, sneezing. Why do those kind of things happen? Because with coughing and sneezing, the antigens which are present inside our body, they are eliminated from our bodies, right? Even running nose. So all those things help us to make sure that the infection is thrown out of our body. And as a response of immune system, what we develop, we develop antibodies. This response given by immune system is stored, and not only this immune system gives or produces antibodies, it also makes sure that the response given by our body to this particular antigen is stored. Right? So immune system also stores its response given to a particular antigen, so that if in future that antigen again attacks, then body has the response to that virus or to that antigen, and thus that antigen can be eliminated very quickly next time. Fine. Stored, and if it comes into contact with the germ again, it recognizes the germ straight away and can start fighting it faster. So अगर next time भी वो ही germ आएगा तो faster response दे सकेगी अपनी immune system, and that's why the response will be stored. Next, so how do vaccines work? That was the work of immune system. So vaccines also try to imitate the response given by our immune system. So vaccines help develop immunity by imitating an infection. Right? So what they will do, they will imitate infection or imitate the work of antigen. They will imitate the work of antigen. This type of infection, however, almost never causes illness. लेकिन अगर एंटीजन को वो इमिटेट कर रहे हैं, लेकिन एक चीज वैक्सीन इंश्योर करते हैं कि उस वैक्सीन से या उस एंटीजन से वो जो यूज किया जा रहा है वैक्सीन डेवलपमेंट में, वो वैक्सीन इलनेस ना दे उस पर्सन को, जिसके अगेंस्ट उसे प्रोटेक्शन चाहिए, वही इलनेस उस पर्सन को ना मिले, ये इंश्योर करते हैं वैक्सीन्स. Vaccines exposes body to the safer version of disease causing pathogens. Why? Because vaccines contain weakened forms of pathogens, और weakened forms of pathogens जब हमारे body में जाते हैं through vaccines, the body gives mild response to those pathogens, and that response is also stored in our memory cells. Right? So safer version of disease causing pathogens, and thereby stimulates immune system to produce antibodies against antigens. It also primes immune cells to remember. This is very important. क्यों? क्योंकि ये response अगर immune system ने store करके नहीं रखा तो वैक्सीन का कोई यूज नहीं होगा. And that's why there is very this step is very important to remember, because in future अगर वो वायरस अटैक करता है तो हमारे पास ऑलरेडी मेमोरी है to develop antibodies against that particular pathogen, and thus that pathogen can be eliminated from our bodies faster. It also primes immune cells to remember the types of antigens that cause infection, that allows a faster response to disease in the future. When the body responds to the vaccine, it builds an adaptive immune response. Right? So this is an adaptive immune response. What is adaptive immune response? This is a response given by our immune system against a specific pathogen. What is adaptive immune response? Adaptive immune response is a kind of response given towards a specific pathogen. Why? Because उस वैक्सीन में वो स्पेसिफिक पैथोजन ही है, और उसके वजह से इम्यून सिस्टम का जो रिस्पॉन्स है दैट इज एडेप्टिव इम्यून रिस्पॉन्स. इट इज नॉट जनरल इम्यून रिस्पॉन्स वेयर immunity is against against pathogens in general. Fine. So it is adaptive immune response. This helps equip the body to fight off an actual infection in next time or in future. Fine. I hope it is clear to you how do they function. So if you have any doubt, kindly raise. If you have any doubt, please raise. Otherwise, we'll be moving to our next part. Fine. So now we'll be discussing about types of vaccines. Types of vaccines. So there are number of types of approaches based upon vaccines are developed. So we'll be discussing about those types based upon what kind of method do they follow for the development of vaccines. So there are several different types of vaccines. Each type is designed to teach immune system how to fight off certain kinds of pathogens or germs. Right? So there are many methods. So what is the focus of scientists while the development of vaccines? So scientists take many approaches to develop vaccines. These approaches are based on, so there are many approaches to develop vaccines, and prime focus of almost all scientists for the development of vaccine is based upon how germs infect cells and how immune system responds to it. फाइन. सो ये जर्म्स जो आने वाले हैं अंदर बॉडी में, अंदर बॉडी के अंदर, तो वो कैसे आ रहे हैं हमारे बॉडी के अंदर, एंड हाउ इज द रिस्पॉन्स गिवन बाय आवर इम्यून सिस्टम टू दोज जर्म्स. नेक्स्ट, हु नीड्स टू बी वैक्सीनेटेड. वैक्सीनेशन किसको करना है? न्यूबॉर्न बेबीज को करना है, या ओल्डर पीपल को करना है, या इन जनरल सभी पॉपुलेशन को करना है? दैट इज ऑल्सो ए थॉट that is there in the during the development of vaccines. Fine. First, germs कैसे body को infect कर रहे हैं, and how our body is giving response against those germs. Secondly, who needs to be vaccinated. Vaccines अगर babies को babies को देने, तो obviously उसके लिए they will have to use other different kind of approach, and for adults they will be using different kinds of approach. Next, regions of the world where vaccine will be used and the strain of virus and environmental conditions there. Right? So, though all human beings have similar genetic makeup, there are certain differences in genes as well, because we have already discussed under DNA fingerprinting that 99.7% of our genes are similar, but 0.3% genes are different. And that is huge number as far as genes are concerned, and hence we also need to focus upon the region for which we need to develop a vaccine. Next, the suitable approach for vaccine development. What will be the suitable approach? Whether we should go for the use of spike protein, or we should use the DNA or genetic material of that virus. That is also being looked into by scientists while development of different types of vaccines. So that is suitable approach for vaccine development. Next, so there are following types of vaccines. So now we'll be discussing about vaccines based upon the approaches that we have discussed. First is live attenuated vaccines. Live attenuated vaccines. So these vaccines contain a version of living virus or bacteria that has been weakened. Live attenuated vaccines. They contain live virus or bacteria, but it is weakened bacteria or virus, so that why it is weakened? So that it does not cause serious disease in people with healthy immune systems. Right? So वो weaken क्यों किया गया है? क्योंकि उससे उसका जो replication है in our bodies, that should be controlled. And if it is controlled, it will not cause a violent response by the immune system of a healthy person who is taking that vaccine, and that's why they are attenuated or weakened. As live attenuated vaccines are the closest to the natural infection. Fine. So this is very important point. This is very important point. These vaccines, live attenuated vaccines, are closer to natural infection. Why? Because they contain the live bacteria or virus, though in weakened form, but it is live, and thus it is closest to the natural infection, and hence they create a strong and long lasting immune response. They create strong and long lasting immune response. Why? Because they are closest to natural infection caused by that virus or bacteria. Right? Example, chickenpox vaccine. Example of live attenuated vaccines is chickenpox vaccine, wherein we are using live but weakened form of chickenpox pathogen. Children with weakened immune systems, for example, those who are undergoing chemotherapy cannot get live attenuated vaccines. क्यों? क्योंकि उनका immune system पहले ही suppress किया होता है, and because of that, if they are getting these live attenuated vaccines wherein live bacteria or viruses are used, then it may be then it may cause there may be some danger associated with live vaccines with people with suppressed immunity, and hence they are not given these kind of vaccines. Next, inactivated vaccines. Inactivated vaccines. So, these vaccines are made by inactivating or killing the germ. यहाँ पे हम क्या कर रहे हैं? हम वो जो germ है उसको either inactivate कर रहे हैं, या it will be killed, means that pathogen will be in killed format during the process of making vaccine. So during the process of making vaccine, that bacteria or germ will be killed or inactivated. Inactivated vaccines usually don't provide immunity that is as strong as live vaccine. So this is very understandable. Why? Because if vaccines do not contain live but weakened form of pathogen, but if they are containing the inactivated or killed form of pathogen, then obviously their response will be lesser than the live attenuated vaccines. So you may need several doses, और उसके वजह से number of doses use किए जाते हैं in order to get ongoing immunity against diseases. For example, inactivated polio vaccine. So example है inactivated vaccine का polio vaccine. Next, subunit vaccines. Subunit vaccine. So what we do here, instead of using complete virus or complete pathogen, we only use a subunit of that particular pathogen for the development of vaccines. For example, instead of using complete coronavirus, only spike protein can be used for the development of vaccine. Fine. So here what we are doing, we are only focusing, or we are only taking a subunit of vaccine, not a complete subunit of virus, not a complete virus. So subunit vaccines include only parts of virus or bacteria or subunits instead of entire germ. As these vaccines contain only the essential antigens and not all the other molecules that make up the germ, side effects are less common. So इसका advantage क्या है? जब हम सिर्फ सब यूनिट या कोई पार्ट ही यूज कर रहे हैं उस पैथोजन का, उसका एडवांटेज ये है कि साइड इफेक्ट्स ज़्यादा नहीं दिखेंगे आफ्टर वैक्सीनेशन. नेक्स्ट, One limitation of these vaccines is that you may need booster dose. लेकिन हम अगर सिर्फ सब यूनिट यूज कर रहे हैं तो इसके वजह से हमें बूस्टर डोज की जरूरत पड़ सकती है फॉर ऑनगोइंग प्रोटेक्शन अगेंस्ट डिसीज. एग्जाम्पल, hepatitis B. You must have heard about hepatitis B. There are several doses of hepatitis B. Especially, I think 2 doses of hepatitis B are given. Why? Because they use subunit vaccine. Because they use only a part of hepatitis B virus. Conjugate vaccines. Conjugate vaccines. So, what are conjugate vaccines? So, there are certain. So, it is for bacteria antigens with an outer coating of sugar-like substances called polysaccharide. So, it is for those antigens, especially bacteria antigens, which contain a sugar coating called as polysaccharides. This type of coating, polysaccharide kind of sugar coating, disguises the antigen, making it hard for young child's immature immune system to identify that okay, this is an antigen. Right? So इसमें क्या होता है for these kind of bacteria, they have the sugar coating which is made up of polysaccharides, and because of this sugar coating, the actual bacteria is getting disguised under that sugar coating, and because of that, if a person's or if a children's immune system is immature immune system, then in that case those vaccines, those bacteria or those pathogens will be able to harm that child's body, and hence what we use conjugate vaccines. Fine. So conjugate vaccines are used against those bacteria or pathogens whose actual genetic material is covered under a sugar coating called as polysaccharides. So conjugate vaccines are effective for these types of bacteria because they connect the polysaccharides to antigens that the immune system responds to very well. So what these vaccines will do? These vaccines will connect these polysaccharides. इन polysaccharides को ऐसे पैथोजन के साथ कनेक्ट किया जाएगा जिसको बॉडी पहचानती है और इम्यून रिस्पॉन्स दे सकती है. फाइन. सो इन पॉलीएस्चराइड्स को, इन शुगर कोटिंग्स को क्या किया जाएगा? किसी ऐसे वायरस के साथ कनेक्ट किया जाएगा जिसे हमारी बॉडी पहचानती है और बेस्ड अपॉन दैट आवर बॉडी कैन गिव इम्यून रिस्पॉन्स टू इट. फाइन. I hope this is bit tricky. I hope you have understood. This linkages helps the immature immune system react to the coating and develop an immune response. Fine. So इसका example Haemophilus influenza type B HIV vaccine is a conjugate vaccine. HIV vaccines are conjugate vaccines. HIV, I am saying HIV vaccines are conjugate vaccines. I hope you have understood. If you have any doubt with respect to this, kindly raise. I'll explain it again. Till that time, let's discuss next one. Toxoid vaccines. Toxoid vaccines. So these vaccines prevent diseases caused by bacteria that produce toxic material. Right? So ये जो toxoid vaccines है, they are specifically targeting those bacteria which leads to production of toxic material whenever they enter inside our bodies. In the response of making these vaccines, in the process of making these vaccines, the toxins are weakened so that they cannot cause illness. So what we use in the development of these toxoid vaccines, we use those toxins developed by these bacteria. See, toxic material means protein only, and if protein is entering, so that toxic protein is if it is entering in our body as a weakened toxic protein, then our immune system will recognize it, and as it is weakened form, it will not harm our body, and that is the mechanism based upon which toxoid vaccines are developed. Right? Weakened toxins are called toxoids. When the immune system receives a vaccine containing toxoid, it learns how to fight off the natural toxin. Booster shots are needed for these kind of vaccines, and tetanus vaccine is a toxoid kind of vaccine. So there is one question. Polyesterides एक बार फिर से. Okay, fine. Polyesterides एक बार फिर से. So see, this vaccine is used for, conjugate vaccine is used for those bacteria. Suppose this is a bacteria, और इस बैक्टीरिया का जो एक्चुअल जेनेटिक मटेरियल है, दैट इज कवर्ड अंडर लेट्स से this blue color material. This blue color material is called as polyesterides. ये ब्लू कलर का मटेरियल है पॉलीएस्टेराइड्स. जब ये पूरा बैक्टीरिया अलोंग विथ पॉलीएस्टेराइड्स कोटिंग जब ये एंटर करेगा किसी के बॉडी में, बिकॉज ऑफ दीज पॉलीएस्टेराइड्स that bacteria is not getting recognized by our immune system. हमारा इम्यून सिस्टम उस बैक्टीरिया को रिकॉग्नाइज नहीं करेगा बिकॉज ऑफ पॉलीएस्टेराइड्स, एंड those bacteria may continue to infect, or they may continue to affect the body of the person whose body is not able to recognize this. Right? So that is dangerous. So what we can do in order to prevent such kind of diseases, we can use, let's say, we will be using this virus in the development of conjugate vaccines. लेकिन हम क्या करेंगे, ये जो वायरस है उसको भी इस पॉलीएस्टेराइड की कोटिंग दे देंगे. इस वायरस को भी पॉलीएस्टेराइड की कोटिंग दे देंगे, एंड देन इट विल बी यूज, और देन इट विल बी अलाउड टू एंटर in the body of a person through vaccines or as a vaccine. जब ये एंटर करेगा किसी के बॉडी में, तो ये जो वायरस है, ये जो ब्लैक कलर में जो वायरस है, दिस वायरस कैन बी रिकॉग्नाइज्ड बाय ए पर्सन्स इम्यून सिस्टम. This black color virus can be recognized by a person's immune system, and because of that, even polyesterides can be recognized as a threat by immune system. And because of this, what will happen, even if these bacteria, even if they are covering or they are coated by polyesterides, as our immune system now learned that okay, polyesterides are dangerous, so even those bacteria which are getting disguised under polyesterides can be detected by our bodies, by our immune systems. Right? So that is nothing but the conjugate vaccine. So what we have done in conjugate vaccine, we have connected polyesterides with a virus which our body recognizes, which is recognized by our immune system. I hope it is clear to you. Fine. So that is about polyesterides. Now next, so toxoid vaccines we have discussed. What are toxoid vaccines? Toxoid vaccines are those vaccines wherein what we use toxic material developed by a bacteria or pathogen itself, but in a weakened form, and that weakened toxin is referred to as toxoid. That weakened toxin is referred to as toxoid. और जब ये एंटर करेगा बॉडी में थ्रू वैक्सीन्स और एज ए वैक्सीन, देन our immune system will be able to recognize them, and thus the immune system can fight against that toxic material, and in future it will also be able to store the response. But we need booster dose for these kind of vaccines, and tetanus vaccine is the example of toxoid vaccines. Next. वैक्सीन का शॉर्ट डेफिनेशन क्या होगा? सी वैक्सीन क्या होता है? वैक्सीन एक ऐसा केमिकल या केमिकल सब्सटेंस होता है जो इम्यून सिस्टम को टीच करेगा. See, that means you did not understand how immune system work. Okay, let me explain this again. इम्यून सिस्टम कैसे काम करता है? इम्यून सिस्टम हमारा जो नेचुरल इम्यून सिस्टम है उसकी बात कर रहा हूँ. सो नेचुरल इम्यून सिस्टम व्हाट हाउ इट वर्क्स. सपोज देयर इज ए अटैक बाय ए वायरस और एंटीजन, then immune system will recognize that antigen as a foreign material, and as it is a foreign material, the immune system will develop a response against that foreign material, so that that foreign material is removed outside our body. Those responses are in the form of coughing, sneezing. Not only this, even in the form of development of antibodies. राइट. सो उस एंटीजन के अगेंस्ट हमारे बॉडी ने एंटीबॉडीज डेवलप की, और एंटीबॉडीज ने क्या किया, उस एंटीजन से फाइट किया और उस एंटीजन को हमारे बॉडी से निकाल दिया. दैट response given by antibodies, or that response given by our immune system in the form of development of antibodies, that response is stored in our immune system, so that in future if there is attack by same antigen again, then our immune system will recognize it faster and will be able to remove it faster from our body. That is how our immune system work. कैसे काम किया इम्यून सिस्टम ने? इम्यून सिस्टम को ट्रिगर करने के लिए हमें एंटीजन की जरूरत पड़ी, राइट. उस एंटीजन के वजह से इम्यून सिस्टम एक्टिवेट हो गए, राइट. सेम मैकेनिज्म हम वैक्सीन में यूज करते, वेयर द वीकेंड फॉर्म ऑफ एंटीजन is put inside the body of a person, and because of that weakened form of antigen, our immune system develops a response against that antigen. See, अगर weakened भी होगा तो भी एंटीजन ही है, तो भी फॉरेन मटेरियल ही है, तो उसके वजह से उसने एंटीबॉडीज इम्यून सिस्टम ने एंटीबॉडीज डेवलप की उस वीकेंड फॉर्म ऑफ एंटीजन के अगेंस्ट. Or not only they have developed antibodies, but also they have stored the response of the development of those antibodies. And if in future same antigen attacks our body, then we have the stored memory of the development of antibodies against those antigens. Fine. I hope you have understood now. Fine, no problem. Next now, so next and the most advanced form of DNA of vaccines are in the form of DNA and RNA vaccines. So abhi I hope you understand what are DNAs and what are RNAs. So instead of injecting a weakened form of virus, they are the most advanced form of vaccines, wherein they do not use weakened form of virus or bacteria into the body as with traditional vaccine. DNA and RNA vaccines use part of the virus's own genetic code to stimulate an immune response. What do they use? DNA and RNA vaccines, they use the genetic code of viruses itself to develop vaccines. फाइन. सो यहाँ पे कोई वीकेंड फॉर्म ऑफ एंटीजन नहीं यूज किया जाता है, या किल्ड लाइव एटेन्यूएटेड फॉर्म भी नहीं यूज किया जाता है. यहाँ पे जेनेटिक मटेरियल और जेनेटिक कोड यूज किया जाता है ऑफ वायरसेस और ऑफ एंटीजेंस इटसेल्फ. And with that kind of genetic material, our immune system is stimulated to produce a response against those antigens. In other words, they carry the genetic instructions for the host cell to make antigens. Right? So they are the most advanced form of vaccines. Right? So the vaccines developed by, let's say, Pfizer or Moderna, they are RNA vaccines. But the only problem with these vaccines is that they need extremely low temperature for storage mechanism, and hence these kind of vaccines are very difficult to use in countries where there is lack of refrigeration as far as vaccine storage is concerned. Okay, next now. Fine. So these are the different types of vaccines. Now, vaccine ingredients. What exactly do we have in vaccines? So we'll be discussing about what are the ingredients of vaccines development. So vaccines use the ingredients they need to be safe and effective. So what kind of ingredients do they use? Those ingredients which will make them effective against that antigen and also safe. Why? Because they should not trigger violent response from immune system, and thus they need to be safe as well. And thus those two conditions, effectiveness and safety, are the prime conditions that are the focus of any organization who is involved in the development of vaccines. Ingredient used in a vaccine serves a specific purpose. Ingredients used in vaccine may help provide immunity against a specific disease, or it may help keep the vaccine safe and long lasting. Right? So again, effectiveness और सेफ्टी, ये दो चीजें प्राइम होती है किसी भी वैक्सीन डेवलपमेंट. So what are the ingredients used in vaccines? Obviously first is antigen. So in order to make vaccine effective, antigens are used. What do they do? As we have already discussed, they teach our immune system how to fight infections, right? Next, so first important ingredient of vaccine is antigen. Second is adjuvants. What are adjuvants? Immunological agent that improves the immune response of a vaccine. So they are the immunological agents which will help increase the immune response of a vaccine. Adjuvants may be added to a vaccine to boost the immune response, to produce more antibodies and longer lasting immunity. So again, this is an ingredient which focuses on effectiveness. Adjuvants, what they do, they make sure that enough antibodies are produced by our immune system, and also the antibodies produced or the response given by the vaccine is long lasting response. So that is the work of adjuvants, and example of adjuvant is aluminum. So these kind of adjuvants help us or help vaccine to make sure that the response given is effective and it is long lasting response. Next, so these are the elements which are used for the effectiveness of vaccines. Next, ingredients to keep vaccines safe and long lasting. So these ingredients protect vaccines from unintended germs from outside. So these ingredients, not vaccines, these ingredients will protect vaccine, and thus they will focus on the safety of vaccine. So which are they? First is preservative. So in order to make sure that the vaccines are preserved for a certain period of time, preservatives are used. Preservatives are generally used in vials, that means those containers of vaccines that have more than one dose. So we know that there are certain vaccines which need more than one dose. तो उस टाइम में और उन vaccines के लिए we need to use preservatives in vials themselves. Vials are those containers. Right? अगर उसमें से सिर्फ half dose ही हम निकाल रहे हैं, तो in order to make sure that remaining half dose is preserved, preservatives are used. That's because every time an individual dose is taken from the vial, it's possible for harmful germs to get inside. Fine. So जब भी हम half dose निकालेंगे उस vial से, तो we are penetrating through needles and then we are extracting that half dose from that vial. So there may be a chance that germs may enter through that opening and they may harm that particular vaccine, the remaining dose of vaccine. Vaccines with single dose generally do not have preservatives. Right? So अगर पूरा ही वैक्सीन उस वायल में से निकालने की ज़रूरत है तो प्रिजर्वेटिव्स का यूज़ नहीं किया जाता है. Right? So they are used for keeping vaccines safe. Next example of preservatives is uh, thimerosa. Thimerosa is a kind of preservative that is used in the vaccines development. Next, stabilizers. Again, this is also focusing on safety component of it. Stabilizers, they focus on safety of vaccines. They help prevent the virus, bacterium, or protein pieces from breaking down and becoming ineffective. They help active ingredients in vaccines to continue work. So their work is to make sure that the active ingredients that are used in the vaccines development or that are put in vials, they remain effective. Right? So thus it is focusing on kind of both safety as well as effectiveness. So they help active ingredients in vaccines to continue work while the vaccine is produced, shifted, or stored. Example of stabilizers is MSG, monosodium glutamate, is used as a stabilizers in vaccines. Fine. So these four are important components of any vaccines: antigen, adjuvants, stabilizers, and preservatives. These four are important parts of or components of any vaccine. There are some additional parts which may be, or additional ingredients which may be there in certain vaccines, like antibiotics. What antibiotics will do in those vaccines? They will make sure that the bacteria do not develop, or if bacteria are developed, they are killed or their problem is addressed. Small amounts of bacteria fighting drugs may be added to vaccine to prevent the growth of germs during production and storage. Right? So they are used to fight germs which may be developed during production and storage process. Cell culture or growth material. अगर हम लाइव एटेन्यूएटेड वैक्सीन्स बना रहे हैं तो वी आर यूजिंग लाइव बट वीकन फॉर्म ऑफ दैट जर्म और पैथोजन. सो उसकी ग्रोथ इंश्योर करने के लिए भी हमें कुछ इंग्रेडिएंट्स यूज करने होते हैं. दे मे बी सेल कल्चर मटेरियल और ग्रोथ मटेरियल लाइक एग्स टू हेल्प ग्रो द वैक्सीन एंटीजेंस. फाइन. सो उन लाइव वायरसेस की ग्रोथ के लिए ये cell culture material या growth material in the form of egg is used. Fine. So that is about vaccines types and ingredients. So if you have any question you can ask, otherwise we will be moving to next part. Fine. So we'll cover this slide till that point. You can ask questions. So disadvantages, we have discussed about advantages of vaccines. Why vaccines are important? They help our body to recognize the antigen which may affect that body in future. Also vaccines help us to prevent diseases. Vaccines help us to prevent outbreaks of diseases, right? So all those advantages or importance of vaccines we have discussed. Also vaccines may help us to fight against antimicrobial resistance. So those points we have already discussed, but there are certain disadvantages of vaccines as well. So what are those disadvantages? Disadvantages are in the form of side effects like allergic reactions that may be developed as a response to the vaccination. Fine. So vaccines are made with different components and these components may cause allergic reactions. So that is one of the disadvantages. Despite getting vaccine, one could still get sick. So this is also possible that, see, vaccines though they are safe, there may be some cases where vaccines do not work. People with compromised immunity cannot be vaccinated or they need medical supervision for getting vaccine. So this is also important point with people who are taking immune suppressing medicines. They cannot be vaccinated. People may suffer from side effects like headache. So side effects of vaccines are there because they are the form of antigens themselves. Fine. So these are the disadvantages, but these are not very serious disadvantages. as far as vaccination is concerned, and that's why there should be no vaccine hesitancy in the minds of people. They should go and they should try they should try get their vaccines, because it will help them to prevent getting infected. Next, vaccine approval stages. So there are number of stages based upon which vaccines are approved to be used in a country. And so generally there are these stages that are being given by Center for Disease Control and Prevention, CDC, of the USA. So these are the stages in the development of vaccine. So first is exploratory stage. We will explore different options for vaccines development, and that exploration will be in the form of research and development. We will try to do R&D, which kind of vaccine we will or which kind of pathogen will be able to use in the vaccine development, or what kind of material shall we use for the development of vaccine. All those things will be contemplated under this stage, exploratory stage, where exploration of different options will be carried out. Next, preclinical preclinical stage, wherein the vaccine which is or the model vaccine will be used on animal models. Right? So जो वैक्सीन हमने डेवलप किया है वो वैक्सीन एनिमल्स के ऊपर टेस्ट किया जाएगा in this preclinical stage. Then it is clinical development stage, and this is the most important stage. Why? Because here in this stage, clinical trials test or clinical development stage, we will be testing vaccines on human beings, and that's why it is the most important stage. There are 3 phases in this stage. So in this stage, trial vaccine is tested on human beings. There are 3 important phases. First is phase 1. So what we'll do in these different phases, see, our focus will be on the sample size. हम phase 1 में कुछ limited लोगों पे वैक्सीन की टेस्ट करेंगे. phase 2 में ज़्यादा लोग होंगे और phase 3 में उससे भी ज़्यादा लोग, right? And also the focus of the examiners will be on different aspects of vaccines in all these phases. So phase 1, a small group of people receive the trial vaccine and the scientist measures antibodies in their blood. So यहाँ पे क्या करेंगे, यहाँ पे small group को ही वैक्सीन मिलेगा और साइंटिस्ट क्या करेंगे, they will focus on the development of antibodies in those people's body. Next, phase 2. In this phase, the clinical study is expanded. That means more number will be given vaccines, and vaccine is given to people with different traits. Not only more number, but also with different traits like age, physical health, etc. Even gender, etc. Here 3 aspects of trial vaccines are assured. यहाँ पे focus क्या होगा? यहाँ पे focus होगा first reactogenicity. Focus will be on reactogenicity, that is ability of vaccine to produce common adverse reactions. So what kind of reactions does vaccine develops, that will be tested in this phase 2 of stage 3. Next, immunogenicity. That means whether it is developing immune response or not, that is also tested under, or that is also looked for in phase 2 of stage 3. And lastly, safety. Is it safe to use? What kind of side effects does it produce? That is also the focus under phase 2. Fine. After this phase 3, here vaccine is given to thousands of people and tested for its efficacy and safety. Fine. So हर phase में third stage के हर phase में हम क्या कर रहे हैं, हम sample size बढ़ा रहे हैं. Initially, let's say tens within means single digit people person को दिया जाएगा vaccine. उसके बाद hundreds of people, and then in the third phase thousands of people will be getting vaccines. Next, regulatory review and approval. So इसके बाद क्लिनिकल ट्रायल्स के बाद बहुत सारा डेटा जनरेट होगा. उस डेटा को हम रिव्यू और रेगुलेटरी अथॉरिटी के सामने प्रेजेंट करेंगे, जो वैक्सीन डेवलप करते हैं वो प्रेजेंट करेंगे, एंड बेस्ड अपॉन दैट डेटा द रेगुलेटरी अथॉरिटी विल टेक डिसीजन whether to approve or not this particular vaccine. Right? So here institutions like CDSCO, the Central Drugs and Standards Control Organization, will review the clinical trial data of vaccines, and based upon their review they either approve or deny use of vaccines. Fine. So इसके इस स्टेज के लिए क्या इंपॉर्टेंट है? इस स्टेज के लिए क्लिनिकल डेटा इंपॉर्टेंट है, जो कि स्टेज 3 में डेवलप हुआ है. In fact, approval for the regulator is required at every stage of these trials. Fine. So vaccines इंट्रोड्यूस करने के लिए, वैक्सीन्स को मार्केट में इंट्रोड्यूस करने के लिए ये स्टेज है, रेगुलेटरी रिव्यू एंड अप्रूवल स्टेज. लेकिन अगर कोई भी वैक्सीन डेवलपर, इफ दे वांट टू इवन गो फॉर क्लिनिकल ट्रायल्स, then also they need approval from CDSCO kind of organizations. Next and last stage is manufacturing and quality control. So once approval is given by the regulatory authority, then the vaccine developer can go ahead with the manufacturing of those vaccines, and they need to ensure that quality is controlled in their departments or in their development units. Fine. So there are these vaccines that are in use. Covishield. First is Covishield, that is developed by the Serum Institute of India based on the AstraZeneca Oxford vaccine. So there was a tie-up between these two, AstraZeneca and Oxford, or Oxford University and Serum Institute of India of Pune. It uses a weakened common cold virus, adenovirus, that causes infection in chimpanzees. Right? So based upon this weakened form of common cold virus, that is adenovirus, Covishield is developed. It has received conditional approval from the CDSCO. Next, Covaxin, developed by Bharat Biotech India Limited (BBIL) in collaboration with ICMR's National Institute of Virology. It is an inactivated vaccine made using particles of COVID-19 virus that were killed. So what does it use? It used inactivated particles of COVID-19 itself, thereby making them unable to infect, and as they are inactivated, they are not able to infect other person or infect the person who is getting vaccine. Fine. It has been approved for restricted emergency use in clinical trial mode with some condition. Fine. So अभी कल परसों ही न्यूज़ था कि उसका क्लिनिकल डेटा अभी मतलब आज भी न्यूज़ है शायद, in today's newspaper only there is news that now the Covaxin developed by Bharat Biotech India Limited, now it is off clinical trials. That means it can be given a approval for use on mass scale. Other important vaccines, as we have already discussed, Moderna vaccine, it is a mRNA type of vaccine and approved in many Western countries. Pfizer, again, it is mRNA type of vaccine, and it was given the emergency use approval by the USA FDA. Fine. So that is about the vaccines. So this was the question that UPSC asked in prelims 2020 about vaccines. So you can read this question and you can answer. Please read and try to answer this question. You have 1 minute with you. Yes, please. So let us discuss this question. What is the importance of using pneumococcal conjugate vaccines in India? Fine. So pneumococcal conjugate vaccines का importance especially in India क्या है ये पूछ रहा है question. These vaccines are effective against pneumonia as well as meningitis and sepsis. So these are the diseases which are pneumococcal diseases, pneumonia, sepsis, and meningitis. And if we are using pneumococcal conjugate vaccine, fine. So I hope you can correlate with the conjugate or the vaccines where polyasterides are used. Right? So this statement is a correct statement. It is used against, it will give protection against pneumonia, meningitis, and sepsis kind of diseases. Dependence on antibodies that are not effective against drug resistant bacteria can be reduced, right? So what we have discussed about an advantage of advantage of vaccines is that our dependency on drugs will reduce, and if those drugs against which bacteria or viruses, if they have developed resistance, then our dependency on those drugs will reduce also. So this statement is a correct statement. Next, these vaccines have no side effects and cause no allergic reaction. So this is a kind of extreme statement that they are making, that there is no side effect and no allergic reaction. So see, every vaccine will have certain kind of side effects. Why? Because it is a foreign material that we are putting in the body of a person, and hence they will have side effects. So this is a wrong statement, and hence your answer should be B, 1 and 2 only. So most of you have given correct answer. Fine. Okay, so this was the question that they have asked. Next now. So we are done with vaccines development. I hope you have understood vaccines development. Now we'll be discussing a last topic of this subject, that is antimicrobial resistance. Fine. So we have came across that term antimicrobial resistance during vaccines development or during importance of vaccines. So with vaccines, if we start using vaccines, then our dependency on the drugs will reduce. And if our dependency on drugs is reduced, then we will be able to reduce antimicrobial resistance. But what exactly is antimicrobial resistance? So let us discuss what is antimicrobial resistance. So antimicrobial resistance, or AMR, is a kind of resistance that occurs when bacteria, viruses, fungi, and parasites change over time. See, as a part of natural process, survival of fittest is the rule, and if and in order to survive for these bacteria and pathogens, if we are using certain chemical against them, then it is natural for them to develop response against those chemicals, and thus to develop a response or to make sure that those chemicals, they do not affect them in future. That is a natural part. That is the natural process of survival of fittest and adaptability. Fine. So over time, those vaccines, or sorry, those bacteria and viruses, they will develop a response against those chemical substances in the form of drugs, and no longer respond to medicines, making infections harder to treat. So what will happen because of this? Because of these kind of resistances developed by these bacteria and viruses, it would be harder for us to treat diseases caused by those pathogens using same medicines in future. अभी सपोज हम कोई मेडिसिन ले रहे और उसके अगेंस्ट बैक्टीरिया ने काम करना बंद कर दिया, तो हम वो मेडिसिन इन फ्यूचर नहीं यूज कर पाएंगे. वाई? Because bacteria is not responding to that chemical that drug now, and hence our dependency on other drugs will increase, or we will need new drugs in order to treat those bacteria, and increasing the risk of disease spread, severe illness, and even death. Fine. So with these kind of features, or with the AMR kind of feature that may be shown by bacteria or viruses, the illness will increase, and there may be the case that in future, that because of non availability of medication, even curable diseases are becoming fatal diseases. That is a danger that we may witness in future. Fine. And that is antimicrobial resistance. As a result of drug resistance, antibiotics and other antimicrobial medications or medicines become ineffective. फाइन. सो इसके वजह से एंटीमाइक्रोबियल रेजिस्टेंस के वजह से वो जो मेडिकेशन है वो इनइफेक्टिव हो जाएगा, एंड इन्फेक्शंस बिकम इंक्रीजिंगली डिफिकल्ट और इम्पॉसिबल टू ट्रीट. What are antimicrobials? Fine. So इसके with AMR the medications will become ineffective, and those diseases which are curable diseases may become incurable in future. Next, what are antimicrobials? So antimicrobials is an umbrella term used for antibiotics, antivirals, antifungal, and antiparasitics. Fine. So it is an umbrella term used for all these kind of medicines. Fine. So antimicrobials are medicines used to prevent and treat infections in humans, animals, and plants. Next, reasons of antimicrobial resistance. ये क्यों होता है antimicrobial resistance, क्यों होता है? First reason we have discussed that it occurs as a natural process of adaptation shown by pathogens. Fine. So it occurs naturally. That is one. Next, underuse or overuse of antibiotics. See, whenever we go to doctor for medication, they give a certain dose. For example, that dose may be of 5 days, 7 days, or even it may be of 3 days, and they ask us to complete that dose. If we leave that dose of, or if we leave that medication in between, then the strains of pathogens which are there in our body, they develop resistance against those incomplete use of medication. Against that medication they will develop a resistance, and because of that, in future that medication won't be able to work against that bacteria or whatever that pathogen may be. Right? So that is underuse of medicines. Even overuse of medications or antibiotics will lead to antimicrobial resistance. Overuse of medication may also lead to antimicrobial resistance. Next, poor hygiene and infection prevention control. So if suppose because of poor hygiene, people are getting sick, and they are getting ill, and because of their illness they are going to doctors, and they are getting antibiotics. So what is the cause of their illness? It is poor hygiene and infection prevention and control mechanism. So if we make sure that the proper hygiene is maintained, and also we focus on infection prevention and infection control, so that many people do not get affected, there is less dependency on the use of medication or drugs, and hence we'll be able to protect ourselves from antimicrobial resistance. But as of now, because of poor hygiene and lack of infection control and infection prevention mechanism, AMR is being seen. Next, misuse in animal husbandry. So we know that in order to increase the production of milk, or in order to increase the production of meat, antibiotics are given to cattle, and also to poultry farms, or they are used in poultry farms. Right? So because of that, those those antibiotics or the medications that are used in animal husbandry, they also enter in human body, and thus that leads to antimicrobial resistance. Fine. Lastly, release of untreated wastewater by pharma companies. See, they also as these are pharma companies, they are developing various antibiotics or various antimicrobial drugs. And if the wastewater that is coming out of these pharma companies, if it also contains strains of those drugs, and if it is released in nearby water body, the microorganisms in that water body, they will develop resistance against those strains of antimicrobial drugs, and those antimicrobial drugs will not be able to cure that person who is taking that medication for the illness. Fine. So that is also a problem, and hence there is need to address this problem. And what are the ways based upon which we can address these problems? Ways are judicious use of antimicrobial drugs. So we should use them judiciously. Rational use of antimicrobial drugs. Ban on the use in animal husbandry. So in animal husbandry, if it is not necessary for the if it is not necessary for the treatment of animals, then these kind of medications should be banned from use. Development of new drugs. See, as we have discussed, AMR is a natural process in the that is exhibited by bacteria. It will happen come what may, and hence there is a need to develop new drugs. Fine. So our focus should also be on the development of new drugs, so that in future, if AMR occurs, then we are ready with new drugs. But right now it is not happening. Many companies are not investing on the development of new drugs, because they are not finding it profitable to invest on new drugs. Why they are not finding it profitable for the investment of new for the investment on new drugs? Because of the patent rules, or because of the TRIPS, because of the trade related with intellectual property rights kind of rules. If they go for R&D, if companies go for R&D, and if they develop new drugs, if they go for R&D and if they go for new drugs, then those new drugs will get patent. The drugs developed by these companies, they will get patent on those drugs. But if that patent makes those drugs unaffordable in different countries, then those drugs can be produced by a country using or as a generic drug, as a generic sorry those drugs can be used by those drugs can be developed by a country as a generic drug, which is allowed under ट्रिप्स एग्रीमेंट. उसके वजह से ये जो इन्वेस्टर्स है या फार्मास्यूटिकल कंपनीज हैं, उन्हें इंसेंटिव अभी नहीं लग रहा है इन द डेवलपमेंट ऑफ न्यू ड्रग्स, एंड दैट्स व्हाई दे आर नॉट गोइंग फॉर द डेवलपमेंट ऑफ न्यू ड्रग्स. But actually, in order to restrict AMR, we need new drugs. Fine. So this is about antimicrobial resistance. So there was one question that UPSC asked in prelims 2019. So please read it and try to answer this question. Prelims 2019 का ये question है. Thank you, Kusum. So kindly read that question, read this question and try to answer, because this is a last slide of our subject. Yes, this is a last slide. Please read and answer. So let us discuss this question. Which of the followings are the reasons for occurrence of multi-drug resistance in microbial pathogens in India? See, UPSC may not use the word that you have read in your PDFs or in your PPTs. Fine, वो कुछ अलग वर्ड भी यूज कर सकता है, बट दैट डज नॉट मीन आपको कंफ्यूज होना है. यहाँ पे उन्होंने वर्ड यूज किया है मल्टी ड्रग रेजिस्टेंस. मल्टी ड्रग रेजिस्टेंस इज सिमिलर टू एंटी माइक्रोबियल रेजिस्टेंस. ठीक है. सो लेट अस डिस्कस these ऑप्शंस. फर्स्ट, genetic predisposition of some people may lead to AMR or antimicrobial resistance. See, this is not at all related. It is AMR or multi-drug resistance is not related with any genetic issue. Fine. So ये एक wrong statement है. Next, taking incorrect doses of antibiotics to cure diseases. Taking incorrect doses of antibiotics to cure diseases. Yes, we have discussed this already, because incorrect dose may include underuse. It may include underuse. Fine. So we have seen that underuse of antibiotics leads to AMR or multi-drug resistance. So this statement is a correct statement. Next, using antibiotics in livestock farming. So we have already discussed that in order to produce, in order to increase production in livestock farming, antibiotics are used, which may lead to multi-drug resistance. Next, multiple chronic diseases in some people. Multiple chronic diseases. See, if there are multiple and chronic diseases, that means they are there for the longer duration, then the dependency of that person on drugs will increase. And if that person is using those drugs for longer time, there are chances that they will develop antimicrobial resistance. That person may develop antimicrobial resistance, and hence it is also a correct statement, and hence your answer should be 2, 3 and 4. Fine. So I hope you have understood this. So fine. So we have done with our syllabus. So just a minute. I'm getting that tab. Just a minute. Okay. So answer is D. So most of you have given answer. So winner सोनू ले आपने क्या आंसर दिया? F ऑप्शन ही नहीं है. विनर सोनूले, क्या नाम है? Navneet, sir, for science and technology, JV study material is enough to prepare. Yes, it is. See, I'll not use one of enough word, but it is a good material. I have seen that material, so you can refer that material, because in UPSC's exam you should not use word enough. ठीक है, वो अच्छा मटेरियल है, आप उस पर रिलाई कर सकते हैं. And supplement it with our classes. Supplement that material with our classes. It will help you a lot. Can you take a special class for remaining important topics before prelims? फाइन शिशिर, I'll मीन्स मैं कमिटमेंट नहीं देता हूँ, but I'll request management to schedule classes, few classes, so that we can discuss important topics. Fine, winner Sonal, no problem. So that's it from my side. That's it from my side. I hope I have done justice with your time, and I hope you have understood this subject. So thank you for your time, and this is our last session, and that's why we are winding up with this session. ठीक है, सो आपको कुछ और पूछना है, बोलना है, तो यहाँ पे बोल सकते हो आप. I'll be there for you for 5 minutes. Thank you everyone. Thank you everyone. Thank you Soumya, thank you Saloni, thank you Kusum, thank you Priyanka. सर 9th क्लास या 10th NCERT पढ़ना चाहिए. सी आई विल सजेस्ट यू टू रीड दोज NCERTs. मतलब 10th तक तो पढ़िए, बिकॉज दे आर इम्पोर्टेंट बिकॉज सी जनरल साइंस से क्वेश्चन बहुत कम आ रहे हैं आजकल, बट स्टिल इफ़ यू हैव सर्टन आइडिया, बिकॉज वेर एवर जनरल साइंस का हमें ज़रूरत था, हमने देखा है. फॉर एग्जाम्पल वी हैव डिस्कस्ड जनरल साइंस फॉर न्यूक्लियर टेक्नोलॉजी, बायोटेक्नोलॉजी. सो जहाँ पे हमें ज़रूरत था हमने देखा है. एडिशनल रीडिंग आप NCERT से कर सकते हैं. Thank you, winner Sonule. There is one question, sir. RVSVV ZEBOV Ebola vaccine is a type of live vaccine. I'm not very sure about that. I'm sorry. See, I don't think it is a live vaccine. Why? Because when they developed these vaccines for coronavirus, these mRNA vaccines, they use the experience of development of vaccine for Ebola. Fine. So ये जो vaccines आए हैं for COVID-19, they are not the first mRNA, or they are not the first genetic code vaccines. They used those vaccines for the Ebola as well. So if that is true, then Ebola का ये जो vaccine आप बोल रहे हो, that is, I think it should be DNA vaccine or mRNA vaccine. But I'm not very sure about this. I'm sorry for that. I'm not very sure about this. Thank you, Navneet. If any problem in revision time, then how we reach you, sir? Okay, I need to share number, right? So this is my number. Oh, sorry. एक मिनट. दिख नहीं रहा है. So this is my number. Fine. So if you have any problem, you can reach me on this number. Yes, Navneet, you can contact me. No problem. Is it important to cover 2019-18 current affairs? See, ऐसा कोई फिक्स्ड फॉर्मूला नहीं है, बिकॉज मेनी टाइम्स वी हैव सीन क्वेश्चंस जो बहुत करंट नहीं होते, लेकिन उन्हें थोड़ा सा उनका मतलब वो टॉपिक्स थोड़े से पुराने होते, बट स्टिल UPSC आस्क्स. सो आई विल नॉट सजेस्ट कि पूरे आप करंट अफेयर्स 2019 18 19 के पढ़िए, बट एटलीस्ट सर्टन आइडिया यू शुड हैव. और वो आइडिया आप कहाँ से ले सकते हैं? दैट आइडिया यू मे गेट फ्रॉम the revision material that you are getting, or the current affairs material that you are getting from JVs are, yes. उसमें उस current affair material में there are boxes in which they have explained certain background of those topics as well. So आप उस पे rely कर सकते हैं. Fine. So with this, I'll wind up this session and our discussion of science and technology as well. Thank you. Thank you for your time. Thank you.